chemotaxonomy of plants is an expanding field of study to seek and seeks to utilize chemical information to improve upon the classification of plants. So we use chemical characteristics or chemical compound to do the taxonomy because we use it when there are no significant or clear biological data to differentiate the plants. So it's also useful in identification of cultivars and hybrids. So when you want to differentiate between certain cultivars, the improvement of the cultivar, for instance, and when you develop the hybrid cultivar, so you can use this chemical compound to differentiate between the varieties, and the use is already used in solving taxonomic problems since the 1960s. So it's not a new science, but it's always expanding. So the plant chemicals also can be used in all classification of hierarchy levels, and of course, uh, it can be influenced by environmental factors such as the temperature. So the secondary metabolites, for instance, they are changing and then are very uh, sensitive to environmental factors. However, it still can be used for the classification. So of course, large and complex molecules may not be advanced than others. So plants which have no taxonomic relationship may probably have the same chemical constituents. So protein contents in plants can change in their lifespan and are different from one organ to another organ. So this is how one of the characteristics, or one why we use the chemotaxonomy in to classify or to differentiate between plants. So again, I will not go into details on the chemotaxonomy. However, in this one, so of course, a large variety of chemical compounds can be found in plants, and quite often the biosynthetic pathway producing these compounds differ in various plants and groups. In many instances, the biosynthetic pathway correspond with existing schemes of classification based on morphology. So in other cases, the results are at variance, thus calling for revision of such schemes. So the natural chemical constituents are conveniently divided into micromolecules, which can be primary metabolites and secondary metabolites, and also macromolecules, which is when we can have a non-cementite macromolecules and cementites macromolecules. The cementites macromolecules are basically the DNA and RNA, so the molecules that carrying the information, which is the DNA, RNA, and sometimes the proteins. You can use this one as cementite, but the non-cementite macromolecules consist of starches, cellulose, and sometimes we can have proteins also. I will give you this just a brief on the discussion on micromolecules and macromolecules. Okay. So of course, the secondary metabolites, metabolomic, we can say this also. This one can perform non-vital functions and less widespread in plants as compared to primary metabolites. These are generally the byproduct of metabolism. They were earlier considered to be waste product, having no important role. However, recently it was realized that they are important in chemical defense against predators, pathogens, allelopathic agents, and also help in pollination and dispersal. This is quite extensive study right now. Of course, it's quite new. We can call it as a metabolomics. I'm also very interested in this particular field. So the following major categories of secondary metabolites are of taxonomic significance. I'm not going to discuss in detail about these compounds. You can read and enjoy the information in the textbook. Please let me know if you have questions. So, of course, the secondary metabolites can conceive or can have what you call as non-protein amino acids. You have can be phenolic groups, which is in the lignin, consists of lignin, flavonoids, and anthocyanins. The nicotine, one of the important secondary metabolites, can be found in the alkaloids group. So hydrogen cyanide can be found in the cyanogenic glycosides, and terpenes consists of one of these menthol and carotenoids. So these are important secondary metabolites that I can give you that usually used and usually found in the plants and or usually used to differentiate plants. Okay. In addition to DNA and RNA, when called as cementite, the information that carry molecules, which under cementite macromolecules, the macromolecules include proteins and complex polysaccharides such as starches and cellulose, which are non-cementite macromolecules. Okay, starches are commonly found in the form of grain, which may be concentric, like in the triticum, in the zea, in the rice, or the or eccentric, like in solanum. And then also this present anatomical characteristic which can be seen under a microscope. These detailed studies of starch grains under SEM also hold promise for taxonomic significance, of course. So in conclusion, chemical evidence has in fact been used ever since man first began to classify plants. When you talk about edible and inedible, it's all about chemical compound, right? If it's edible, which means that it has a compound that good to eat, safe to eat, unlike the inedible. So obviously this is based on their chemical differences. So these are, we can say that chemical evidence or chemical human use chemical to differentiate plants. So this is one of the history or the origin of the classification. So, and of course after that, human also look at chemical information about medicinal plants, like in herbs, as another example. This was concerned with localization and application of physiologically active secondary metabolites such as saponins and alkaloids. So basically, when you have plants that you have, you can say that, oh, this plant can be eaten, or this one is cannot be eat. Then you can say, or then after that, probably you can use a morphological characteristics to educate other people to say, oh, these types of plants can be eaten or not, or this one can be used for medicinal plants or not. Therefore, plant chemicals can be used in all classification and in all classification of hierarchy levels.